welcome to Biology, the scientific study of life. Evolutionary adaptations of vertebrate digestive systems are often associated with diet. The digestive systems of mammals and other vertebrates are variations on a common plan, but there are many intriguing adaptations often associated with the animal's diet. We will examine just a few. Some dental adaptations, dentition in animals, assortment of teeth is one example of structural variation affecting diet. Particularly in mammals, evolutionary adaptations of teeth for processing different kinds of food is one of the major reasons the vertebrate class has been so successful. Compare the dentition of carnivores, herbivores, and omnivores mammals in this figure. Non-mammalian vertebrates generally have less specialized dentition, but there are interesting exceptions. For example, poisonous snakes such as rattlesnakes have fangs-modified teeth that inject venom into prey. Some fangs are hollow like syringes, while others drip their poison along grooves on the surface of the teeth. All snakes have another important anatomical adaptation associated with feeding. They swallow their prey whole with no chewing, and the lower jaw is loosely hinged to the skull by an elastic ligament that permits the mouth and throat to open very wide for swallowing impressively large prey. Stomach and intestinal adaptations. Large expendable stomachs are common in carnivores, which may go for a long time between meals and therefore must eat as much as they can when they do catch prey. For example, a 200 kilogram African lion can consume 40 kilograms of meat in one meal. The length of the vertebrate digestive system is also correlated with diet. In general, herbivores and omnivores have longer alimentary canals relative to their body size than carnivores. Vegetation is more difficult to digest than meat because it contains cellulose. A longer tract furnishes more time for digestion and more surface area for the absorption of nutrients. Symbiotic adaptations. Herbivorous animals face a nutritional challenge. Much of the chemical energy in their diets is contained in the cellulose of plant cell walls, but animals do not produce enzymes that hydrolyze cellulose. Many vertebrates, as well as termites, whose wood diets are larger cellulose, solve this problem by housing large populations of symbiotic bacteria and protists in fermentation chambers in their alimentary canals. These microorganisms do have enzymes that can digest cellulose to simple sugars and other compounds that the animal can absorb. In many cases, the microorganisms can also use the sugars from digested cellulose along with minerals to make a variety of nutrients essential to the animal, such as vitamins and amino acids. Location of symbiotic microbes in the digestive tracts of herbivores varies depending on the type of animal. The hoatzin, an herbivorous bear that lives in South American rainforests, has a large muscular crop and esophageal pouch that houses symbiotic microorganisms. Hordes in the wall of the crop grind plant leaves into small fragments, and the microorganisms break down cellulose. Many herbivorous mammals, including horses, house symbiotic microorganisms in a large cecum, the pouch where the small and large intestines connect. The symbiotic bacteria of rabbits, and some rodents live in the large intestine as well as in the cecum, as shown in this figure. Since most nutrients are absorbed in the small intestine, nourishing byproducts of fermentation by bacteria in the large intestine are initially lost with the feces. Rabbits and rodents recover these nutrients by eating some of their feces and passing the food through the alimentary canal a second time. The female rabbit pellets, which are not reingested, are the feces eliminated after food has passed through the digestive tract. The koala, an Australian marsupial, also has an enlarged